Hi, welcome to Moreauknows. Today we're going to be learning about the Biology Essential Standard 1.2.1. We're going to explain how cells use buffers to regulate pH and how cells can respond to maintain temperature, glucose levels, and water balance in living organisms. We've already talked about water balance through osmosis in a previous video, so today we're just going to be focusing on how living organisms maintain a balanced and internal and external environment in our world. And so the first thing you're going to need to know is homeostasis, and that's the regulation of metabolic processes that maintain a stable internal conditions that are required for survival. That's a long definition. Basically, if we have homeostasis, we want things to stay the same. The first thing we have to look at is the pH, how we maintain pH in our body. Now our blood pH level is about 3.5, and we really need to figure out what that means. And so the pH scale is something that we look at to see how much hydrogen ions that we have in our blood, or OH ions if we're in base. The pH scale determines how acidic or how basic something is. And if we look here, we have bleach. That is a very basic solution, and so it has a really high pH value. And if we looked at something like our stomach has an acidity of 1, and that's a very acidic solution. And if we have something like distilled water, that's going to be about a 7. Right here at about 7.35, we're going to see our blood pH level. And if it gets too high or gets too low, it's really bad for us. We can actually die because our enzymes won't work. If we want to identify acids and bases, we need to know some things about them. And so acids have a pH value from about 0 to 7 on the pH scale. The lower the pH value indicates a stronger acid. So it's kind of weird when you think about 0 being lower being a stronger acid, but that's what it is. Bases, or another word for them is alkalines, have a pH from about 7 to 14. So the higher the pH value indicates the stronger the base. Well, what if we have something that's exactly at 7, right in between acidic or basic? We have something called a neutral pH, and that's going to be around 7, and that's going to be considered a neutral substance. Our blood is about 7.35 on the pH scale, and it really needs to stay there. If it goes too high or goes too low, we can die because our enzymes won't work. They'll denature. So how do we maintain a pH of 7 when we are eating foods like orange juice and lemon juice and maybe consuming some basic solutions such as milk or coffee? We need to keep our pH balance inside our cells the same. So our body has many different kinds of buffers, and a buffering solution is just going to maintain that pH level up to a certain degree. So if we get too high or we get too low, yes, it is going to change, but it's going to take a lot more acid or base to change that pH level. A buffer solution is a solution that changes in pH only slightly when adding small amounts of a strong acid or a strong base. And so we'll be able to use this in the lab to determine a buffer. If I was to determine what a buffer does on a chart, well, if I have water and just water, and I added, and the pH of water is at a 7, and I added OH molecules, which is in something like NaOH, or sodium hydroxide, the OH molecules are basic. And so because it's a base and we add it just to water, it increases in pH value. So it increases to become a base because we're adding base. In the opposite effect, if we added hydrogen ions, which can also be written as H+, and the one that we're going to be using is H2SO4, which is sulfuric acid, and these hydrogen ions in water will dissociate. and they'll be included into the liquid and make the water more acidic. And so if that happens, the pH value will lower, making it a stronger acid. So the closer that we are to zero, which we'll never get to zero, but if the closer we get to zero, the lower the pH value. But the stronger the acidity is for that solution. Now let's look at a buffer in the same scenario. Well, if I have a buffer, and let's say it buffers around 4.9 or 5, somewhere around there, we can add hydrogen ions so we can make it more basic, but it's not going to change very much. It's going to stay the same. And in the same aspect, if we add an acid to it, the same amount that we would add to water, it doesn't change as much. And so we stay around the same pH value even though we're adding acids and adding bases, which in water would normally change the pH dramatically, either higher or lower. So how can we indicate what the pH of a solution is? We can use something called an indicator. And an indicator will actually change colors in specific ranges of pH levels. And if we use these, we can determine exactly what the pH level is just based on the color. And so some of the ones that you might use later on in your life might be bromethanol blue, phenol red, and phenolphthalein. And so they change colors dramatically from a white at the pH of 8 to a pink at the pH of 10 to tell you what the pH level is of that solution. Not only does our body regulate the pH value in our blood, but we also got to regulate the temperature of our blood and our cells, and so do all the other living organisms. But in humans, we have something called thermoregulation, and it's our body's attempt to maintain an internal temperature, and we're trying to maintain that homeostasis. So we have something called thermoregulation, and I know we've talked about this in class, but you might shiver if you're cold, and that's going to excite your muscles and create some heat. Or if you're too hot, you might sweat, and that would try and cool off your body by using water. There's some other ones that we've talked about in class, but those are the two main ones. Along with temperature, we need to regulate glucose. And glucose is stuff that we eat, and it's a carbohydrate, that's a sugar, and we know that because of the OSE here at the end of the word. And the regulation of glucose takes place in our bloodstream. And our body, our pancreas, produces insulin. And this will allow us to regulate the amount of blood glucose levels in our body. If we cannot produce insulin, we would be considered diabetic, and we would need to take insulin from an external source to regulate this amount of glucose into our body. Thank you for watching Moreauknows. Now you do.